So, can everyone hear me? Can you hear me? Can you hear me? Hello? Hello, everyone? Can you write yes, ma'am? आवाज़ आ रही है ना मेरी राहुल? यस यू कैन हियर मी राइट? आवाज़ आ रही है ना मेरी? आवाज़ आ रही है? ठीक है, शुरू कर दूँ फिर? जस्ट 2 मिनट का ब्रेक दीजिए, फिर मैं शुरू कर देती हूँ। ओके, राइट, तो इसको सो yeah, so it's going fine. I just need 2 minutes. So now what do you see? Nothing, right? So I would need 2-3 minutes to finish this. Now I just have a delay of 2 minutes and then I'm starting, right? So I have 29 plus. चलिए, आपको दिख रही है स्लाइड सब? कैन यू सी दिस पिक्चर्स? कैन यू सी दिस पिक्चर्स प्लीज? चलिए, so can you see these pictures? Yes, Rahul. Right, sorry, there is a disturbance, that's why I thought maybe I can take it some other time, but yeah, done. So just give me 5 more minutes and I'll be through. 5 more minutes, you can plan and organize yourself. Just 5 more minutes. Right, and now what is this? Yeah, scene 2 is this. Yeah, and scene 3, yeah, and scene 4. क्लिक प्लस बटन बिलो. कहाँ है प्लस बटन बिलो? उसको फुल स्क्रीन कर देता है. क्या चीज़?
बेटी की शादी है।
अच्छा, जरूर जरूर आएंगे। अच्छा, ये है नहीं घर में क्या?
ऐसा चलेगा तो मोबाइल वहीं रहेगा, दुकान पर रहेगी, मिक्सर ऊपर काम करके निकल जाता है।
अच्छा, ठीक ठीक है, जरूर आएंगे। Right. So do we start now? Let me do it now. Chaliye. So I hope it is okay and all of you can see me now. Can you see me now? I will start as soon as a yes comes from any one of you. Voice nahi aa rahi hai. Is it? Can you hear me now? I am starting it now. Do I write this on this thing? So is it going fine? You can hear me? Aman, can you hear me? Aman Chaudhary. Yes. Can you hear me, Aman? Yes. So it is done. Sorry, I started now and I am teaching you what? A very interesting topic, that is linkage and linkage mapping. And gradually as you go through, you will find that it is one of the very important topic in the genetics and a very interesting one, and this would be far above what you have studied in the schools. So it was worth for me also. I thought I didn't want to do it in a hurry, so I took my time, but still again it was a very confusing and a very busy day for me. So I somehow have tried this time. We thought we'll do it at 8:30, but still 25 minutes again gone in one thing or the other happening around. But now I think I am ready to go. So do we start? All of us have come. Rahul, you want to make an announcement that it is starting and everybody should come now so that they don't miss it, right? So let's do it. I begin with this, and the first thing here is I am teaching you what today is linkage, recombination and sex linkage maps and sex linkage. But let's see how long it goes, because I will not be able to take it for very long. So let's see till how long we can do it, right? So what we'll be studying in, can you, all of you are able to see this, right? You can see what we'll learn today, this slide. Neha wrote last. Neha, can you see that what all I am going to teach you today? Neha, Mansi, all of you have been writing, so I hope you can see this now. You can see, right? Sure. There's some communication internet problems, but I will carry on and I will not stop myself now, and I'll keep going, right? So we'll be talking about what is linkage, and we'll know the discovery of linkage. So I basically have tried to cover what is there in your syllabus in NEP or DSC paper for BSc Life Sciences. So we'll know what is linkage and how it was discovered, and you will understand it has been quite interesting. And then we'll learn how to draw a linkage map, and we'll perform few 1 factor, 2 factor, 3 factor crosses, because as it is given in your syllabus in this form only. Then we will talk about sex linkage with the help of examples of eye color and Drosophila, color blindness and hemophilia in human beings. But I am doubtful we'll be able to reach there or not, because it has become quite a lengthy one, and I hope you'll enjoy this. So let's begin with linkage. First, what is linkage? Linkage is the tendency of or the nature of the genes which are located on one chromosome to be inherited together. What does that mean? That in one chromosome we have several genes, and for you I have arranged an example. I have found an example of this human genome. Maybe we have 23 pairs of chromosome and we have 35,000 genes. Now the number is 30,000, gradually it is coming down, and on average if we see we have 1,500 genes located on one chromosome, right? So you can keep asking wherever you want to. And we have 1500 genes located on one chromosome, and all of them try to go together. But how? We'll understand that how they want to go together, with whom, which can go together. And but obviously they will all are not independently assorting. So how this thing is happening, that you have 1500 genes on one chromosome, average, this is an average figure, and how these are managing each other, that we would know. Is it okay? Then we go next. But I told you the definition of linkage. And linkage is the tendency of genes located on one chromosome maybe to be inherited together. And opposite of just linkage, what is opposite of linkage is recombination. Is linkage is that they are located on one chromosome, but something is happening to that the new combination comes every time. And how this combination comes is by a process, all of you know, is crossing over. And the crossing over is what? Exchange of parts during meiosis prophase, all of you know, and I will not go much beyond the definition of this. And now what is this? Is that linkage would try to keep the parental genotype, and crossing over will do what? Will try to separate them and give you the output of what? Recombinant chromatids or recombinant phenotype. Okay with all these things? And what is happening in nature is that you don't get complete linkage, or no, meaning you don't get that, it doesn't happen that you always have a parental. That is why you see around you have so many combinations, because of these 30,000 genes we have, they are located on 23 chromosomes. They keep randomly arranging and going to the sister chromatids and here and there. So I hope you got these three definition of these three terms. And right, so let's go to the next one, and let's begin with the linkage in more details. So if you have, we talk about two genes in a cytoplasm, maybe in a nucleus. What happens is that we're talking about two, I hope you can see this picture here. I have shown you that I drew in the class also, that by independent assortment, what happens if the genes are located on two different chromosomes. Just try to understand. Here I am talking about the chromosomes and gene A and B. So what will happen? The things which will happen is what you have studied in independent assortment, meaning they will assort independently, and you will get all four type of ratios in the equal proportion, right? You don't get the parental type more, you get all four in different proportion, in the equal proportion, right? And if we look at the other one, that if we have linkage, what will happen if we have linkage? If we have linkage, you will see only the parental type, right? AA is parent, and small aa is at the other side. AB is the parental type. So you see either capital A, capital B, or small a, small b. So if it is linkage, then you will have only parental type. If it is independent assortment, you will get what? Equal proportion of all four, right? So let's understand things with the help of one experiment, is on Drosophila. And this is, what we're talking about here is the Drosophila, and we are studying which character? BW and HV. BW plus means what? It refers to the wild type. So if we write only plus, that means it's a wild type, and wild type is always dominant, right? So if wherever you see plus, it means it's wild type and it is dominant, right? And so if you want to know the details, which phenotype these characters carry, you can ask questions wherever you want to. Voice aa rahi hai na sabko? Are you able to hear? Anjali is writing that she cannot hear me. I hope all of you can hear me, right? Okay with this? Yeah, I think so. If you have any problem, you can write. So HV is heavy wing vein. आप देख रहे हैं ये wings में veins दिख रही हैं. I hope you can see. And BW is brown eye, right? Brown eye is mutant, and heavy vein also is mutant, and the normal wild type is thin winged veins and red eyed, right? So this is normal type, plus is thin vein and red eye, and this is the mutant type. So these two characters we are studying, and what do we observe is, in the cross one, when we see that there is no crossing over happening, what will have? You will have only the parental type, right? You will have only the parental type. And if you have a test cross, in the test cross you cross this with the homozygous recessive, and what do you get in here is, assuming that there is no crossover, in the second generation what happens is you get a ratio of 1:2:1, and in the test cross you will get a ratio of 1:1. So you get a ratio of, please note, you get a ratio of 1:2:1. If this would have been independent assortment, what would have you got is, can you write 3:1, not 1:2:1. Phenotype I am talking. So in case of linked genes, you get a ratio, meaning 1:2:1, and if it's a test cross, you get a ratio of 1:1, right? So if it is a complete linkage, right, you will get a ratio of 1:2:1, phenotypic and genotypic ratio, and in the test cross you get a ratio of 1:1. This is linkage ratio. This does not happen if it has been a Mendelian inheritance, right? So let's come back to the discovery part, how this was discovered, and we will discuss the several crosses which were made by Morgan. Thomas Hunt Morgan, what is he known for? He discovered sex linkage. During the process of studying, linkage, he encountered several things which were new, and he in this process also discovered this sex linkage. He was the first one, because his contribution comes in the exam, you might be asked. He was the first one to propose the term crossing over. Then he also said that linked gene can also get separated by what? By crossing over. And then he said that frequency of this separation depends upon their location on the gene, meaning how far they are on the chromosome. That is how they are separated from each other. This was Morgan, Thomas Hunt Morgan, and his contribution comes in the exam very often. Last time also it came. So let's talk about the Morgan's experiment. What did he do? I have shown you the photo of Drosophila, which is, is showing you what is the phenotype which he has been studying. So which phenotype or which characters Morgan studied? What? So which characters Morgan studied were, first is wild type was gray body, right, which he has written with, and red eyes. Red and yellow is the character, and the mutant is yellow body and white eye. So W is for eye, Y is for yellow. So we have gray or yellow, white or red eyes, right? This was the wild type. And for cross A, in the first cross, what happened is, when he crossed yellow and white female to wild type, what did he observe? That all the females in the next F1 generation were wild type, right? He thought some error, something unique has happened. He reversed the cross, meaning he took other characters, is W and M is miniature wings. There was another character which was miniature, and studying these two different characters, then you would know that there was something, which he discovered, just because when he thought that this may be because of error, maybe these characters are unique. That is why it is happening, right? So, sorry. So this was it, right? So he made two crosses. Cross A was what? where he had wild type was grey body and red eyes, and cross 2 was what? Normal wings and red eye. And what did he observe in both the crosses is that F1 female was always wild type, right? This is what he observed, and F1 male observed both the characters. So with this experiment he found that something different is happening in these crosses, and they are not following the Mendelian inheritance, because sex also plays an important role in inheritance, sex linked inheritance. That is how he reached to sex linked inheritance. And when he did this cross in F2 generation, what did he observe? That was very surprising to him. He observed a ratio of 98.7% showed the parental phenotype, and the recombinant were only 13%. In cross A, what did he observe? The parental were 98.7, and recombinant were only 1.3%. This was cross A. And in cross 2, what did he observe? That 62.8% showed the parental type, and 37.2 showed the recombinant type. So because he was doing 2 crosses and observing the same thing, like we talk about these reasons for success of Mendel. So what do you say? What is the reason for success of Morgan? What did you observe? I hope you are listening till now. Any answers you want to write? What was the reason for success of Morgan? That the crosses he performed, what did you do? Right? I'm waiting for your answer because I need a break for 2 minutes. I'm done with half maybe. But the interesting crosses are still remaining. So anyone wants to tell me what was the reason for success of Morgan? I have told you this already. I am waiting for the answer. Right, I was here. कोई लिख रहा है? Yes, let me see how many yes ma'am comes now. If you are there, you will write yes ma'am, I'm sure. If you are listening to me. It takes 2 minutes for response to come. 1 minute or 30 seconds. चलिए आते रहेंगे जवाब और आगे चलते हैं फिर. So this was it, that he observed parental ratio in much higher number in both the cases. That is how he discovered that something is wrong. The Mendel's law of independent assortment is not being followed 100%. Right? Then we go forward to his student, that is Strutwent, Alfred H. Strutwent, and his contribution also comes in the exam very often. That you will be asked to write about the contribution of Alfred H. Strutwent. Now, what did he do? He said that the crossing over frequency could be used to map the sequence of genes and to find out the distance between 2 genes. So what was that? What did he do? He compiled the data of these crosses, and then I hope you can hear me. Yes, Sujal, good. Can you hear me? First of all, write an answer to this. In a cross, genes are located near. That's why parental type shown due to linkage between them is there. But in a cross B, there is a distance between the 2 genes, right? That is why the recombination occurs. But see, this cross I'm talking, that is why you are starting from the other end, right? Because we didn't know earlier, he was in the process of discovering that something is happening. The law of independent assortment is not being followed, and the linkage, the phenomenon of linkage is there, right? Your answer is right, but if you know the things. But I asked you what is his reason for success of Morgan, because he compared the two crosses at the same time, and then he realized that there is something happening, that there is a difference there is a difference in the phenotype according to sex first of all, and the location of the genes also matters, meaning law of independence assortment is not being always followed, right? So what did Strutman do? Strutman told is that there is this recombination frequency, what he gave just now, in 98% or 93%, that can be used to plot and on a gene map, and to find out the distance between the genes. This was Strutman who first gave this concept. And then what happened? He, for example, said that if there is a recombination frequency of yellow and white is 0.5%, that means they are distant at 0.5 unit, right? And he said when we make different crosses, we can sort out for all the genes. Like in this case, he said yellow and white is 0.5, and then white and miniature is 34.5, and then he said yellow and miniature is 35.4. That means the W should be in between. This is how he said we can sequence the complete chromosomes and know which gene is closer to this one by making different crosses. Then he gave example of one more cross. This is how he explained that how we can arrange genes on the chromosome. So Strutman said that the percent recombination can be used to plot the genes on a chromosome, and secondly we can also know their sequence also, right? Then he further added many things in here. Is like he said one map unit is equal to 1% recombination between the genes. Linkage also occurs in autosomes. It's not a phenomenon which is restricted to sex chromosome, but they said in Drosophila crossing over occurs only in females, because they were mostly studying the genes located on sex chromosome. That is why they said it only occurs in females. Then crossing over occurs at the 4 standard stage, and single crossover between two non-sister chromatids other than two chromatids will be unchanged. And he said rarely you get 100%, and meaning, right, so for single crossover the maximum percentage you can get is 50%. These few more points he pointed out. And then he also gave you the concept of multiple crossovers. You got it? Because there was a randomness still observed, because the location and the distance on chromosomes was not completely answered. They still observed some randomness, and that is why they said that maybe there is something else is happening. So Strutman gave this multiple cross concept also. So can you believe that if for this gene double crossover, what is happening? Because of double crossover, only B is being exchanged. So he gave this concept of multiple crossover, which brings some randomness, that the percent frequency is not able to answer everything, right? So he said the probability of double crossover between A and C can also be calculated by multiplying them and make a correct gene map. So he gave the concept of multiple sequences and multiple crossovers, and he said that we can do it this way. The other thing is that there's, just hang on 1 minute, genotype all the offsprings and the population, right? So this is what he told. Another thing is that right, so we have to leave it here. This was, so with this one I could explain you what is, this is the reference book. Here it is given the best. So of course the lecture was fine, you can study it from here also, but still if you want to study more, I think it makes sense for you to study it from the this book. That will actually enhance your knowledge, because I had my limitation. I could not take you to the depth because of the time limitation again, and I could not do this way, but still a lot of syllabus is remaining, so I might do it this way in one or two more topics. But for now I think I have done whatever was written in your syllabus, but sex chromosomes is still remaining, sex linked inheritance, that I might take in the offline class on Friday, and then I will go to the next topic. But still I'll be very happy to answer your questions and interact with you if you want to know anything or ask anything. Anyone with any questions? Yeah. Abhishek, Avantika Parmar, Vinita Arya, Aman Chaudhary. You have any questions? Please ask me if you want to. So how many genes are there on human chromosome? I will ask you. How many genes are? I was trying to write the recap, but I didn't have time. How many genes are present on human chromosome? In human genome. Number of genes in human genome. Number of genes. अच्छा ये बताइए, क्लग एंड कमिंग में से प्रिंटेड नोट्स किस किस को चाहिए? आई स्टिल कैन गिव यू. देखिए मैं आपको बताती हूँ. I studied this in BSc honors. Then after that I was teaching this to BSc students for many years. Then this paper gradually went to zoology. So then we were disconnected, but now still I am enjoying this a lot to teach you this paper. So when there are no questions, still I would love to answer if you have any questions. Average, it's not average, I tell you Sujal. The average, this is not average number of 30,000. What is happening is that on the basis of bioinformatics studies, people predict that this may be the number of genes present. But as our studies are getting refined, the number is getting reduced because there is a lot of redundant DNA in everyone. So the number of genes, that is why keep coming down, right? Earlier they projected 35,000, now it is 30,000, right? So who gave the concept of chiasmata? Chiasmata. What is chiasmata? Can you answer? Anybody wants to answer what is chiasmata? एग्ज़ाम में आएगा ये सब, मैं बता रही हूँ. पिछले साल का क्वेश्चन पेपर आपको चाहिए तो मेरे से ले लीजिएगा, एंड आई कैन डिस्कस दैट ऑल्सो इन द क्लास. लास्ट ईयर्स पेपर, राइट? So when there are no more questions, I think I need to wind it up. सेक्स लिंक्ड इनहेरिटेंस मैं आपको फ्राइडे को क्लास में करा दूंगी, और उसके बाद कुछ एक टॉपिक्स और रहते हैं, then I might have to do them this way only. Is it all right with everyone? I think I need to leave now and stop this class here.